12th Standard Bio-Zoology Chapter 5 Molecular Genetics Process of Transcription Video 2. In the previous video, we have already discussed something about transcription. In this video, we are going to learn the remaining part of transcription. Transcription is a three-step process. It involves initiation, elongation, and termination. How can the RNA polymerases control the three-step activities? This is a question. How the RNA polymerases are able to catalyze the three steps. RNA polymerases can catalyze the elongation. RNA polymerases can directly catalyze the elongation. RNA polymerases associate with sigma factor to cause initiation. RNA polymerase directly involves in elongation. For the purpose of initiation process, it combines with sigma factor, there is temporary association with sigma factor. RNA polymerase has temporary association with termination factor, rho factor. Because of the association, temporary association, the RNA polymerase is able to control the termination. RNA polymerase is able to control initiation and termination also because of the temporary association with the sigma factor and the rho factor. The sigma factor initiation factor and the rho factor termination factor, both the factors instruct the RNA polymerase for initiation and termination. At what time initiation must happen and what time termination must happen is instructed to the RNA polymerase by the sigma and rho factors. The diagram shows the association between sigma factor and RNA polymerase. This diagram, our rho factor is inside the RNA polymerase. Now we can understand how the RNA polymerases are able to catalyze the three steps in transcription. Transcription in bacteria is different from eukaryotes. In eukaryotes, there is separate compartment called as nucleus. The genetic material DNA is separated from other organelles by nuclear membrane. Transcription takes place in the nucleoplasm. After transcription, the mRNA comes out and it comes to the cytoplasm. In the cytoplasm, protein synthesis takes place. That is, translation takes place in the cytoplasm and the transcription takes place in the nucleoplasm. There are two separate places for transcription and translation in eukaryotes. In prokaryotes, the mRNA does not require any processing to become active. We will learn at the end about the processing. The mRNA formed from the DNA is not a functional one in eukaryotes. It needs three steps processing. We will study at the end. But the prokaryotes do not need the three steps processing. Transcription and translation take place simultaneously in the same compartment in prokaryotes. We have already learned in eukaryotes there is separate place for transcription and translation. In prokaryotes, transcription and translation can occur simultaneously because both occur in the same compartment. Transcription can begin much before the mRNA is fully transcribed. In bacteria, before the mRNA is fully transcribed, translation can begin. Translation can begin when the mRNA is partly transcribed in prokaryotes. But in eukaryotes, after the mRNA is fully formed and it is processed, it comes out, then only translation, that is protein production, take place. Now let us see the transcription in eukaryotes. RNA polymerases are found in organelles. RNA polymerases are also found in the nucleus. Both are necessary for transcription in eukaryotes. Transcription in eukaryotes needs RNA polymerases found in organelles as well as RNA polymerases found in nucleus. At least 3 types of RNA polymerases in nucleus are necessary for transcription in eukaryotes. RNA polymerase 1, it transcribes rRNAs. It transcribes 28S, 18S and 5.8S RNA polymerases. S is a Svedberg unit. It defines the size of the small particles, size of the RNA. And RNA polymerase 3, it catalyzes the transcription of tRNA, 5S rRNA and snRNA, small nuclear RNA. RNA polymerase 2 plays a significant role. It transcribes precursor of mRNA. The precursor of mRNA is the hnRNA, heterogeneous nuclear RNA. It is called as heterogeneous nuclear RNA because it has introns and exons along the length. Introns and exons are arranged in the newly formed mRNA. So it is called as heterogeneous nuclear RNA. The 3 different kinds of RNA polymerases play 3 different roles. There is division of labor among them. So the 3 are also necessary for the formation of or for the transcription of RNAs in eukaryotes. Eukaryotic genes have exons and introns. Because of the exons and introns, the newly formed mRNA is heterogeneous. The exons are expressed sequences. They are coding sequences. They can code for protein. Introns are intervening sequences or non-coding sequences. They cannot code for protein. So the introns have to be removed to make the RNA functioning. Now let us see. The removal of introns is called as splicing. The introns are removed by a process called splicing. At the 5' end, methyl guanosine triphosphate is added. At the 5' end of newly formed RNA, methyl guanosine triphosphate is added. This process is called as capping. Capping involves adding of methyl guanosine triphosphate at the 5' end. At the 3' end, adenylate residues, 200 to 300 residues are added at the 3' end. This is called as tailing. Tailing involves addition of adenylate residues at the 3' end. Capping involves addition of methyl guanosine triphosphate at the 5' end. Removal of introns is called as splicing. After the completion of the 3 activities, the heterogeneous RNA becomes functional mRNA in eukaryotes. It shows exons and introns. This diagram shows exons and introns in mRNA. The exons are yellow in color and introns are brown in color. At the 5' end, methyl guanosine triphosphate residues are added. This is called as capping. At the 3' end, adenylate residues, poly A tail is added. This is called as tailing. Then the introns have to be removed. The introns are removed by splicing. The introns form a loop and get detached. This is called as splicing. Capping, splicing and tailing makes the heterogeneous nuclear RNA becomes functioning one. Split gene feature is found in eukaryotic genes. What is split gene? Presence of introns and exons. Exon 1 intron, exon 2 intron, exon 3 intron, then exon. Introns and exons are arranged. This is called a split gene feature. After splicing of the introns, the 4 exons are obtained. The 4 together form an mRNA. This is normal splicing. Alternative splicing is also there. Now let us see what is alternative splicing. When the splicing cut is here, this intron and exon 4 will be removed. This intron and exon 4 will be removed. Then what will be remaining? Exon 3, exon 2 and exon 1. So alternative splicing produces particular kind of mRNA. It includes exon 1, 2, 3. It produces a particular protein. Let us see another alternative splicing. If the splicing takes place here, the exon 3 will be removed along with 2 introns. So the mRNA will contain exon 1, exon 2 and exon 4. Exon 1, 2, 4. Another protein will be formed. This is alternative splicing. Alternate splicing patterns is a mechanism known to play an important role in generating both protein and functional diversity in animals. Variety of proteins and variety of animals are formed because of the alternate splicing pattern. Introns would have arisen before or after the evolution of eukaryotic gene. We do not know that. If introns arose late, how did they enter eukaryotic genes? If introns arose after the evolution of eukaryotes, how did they enter the eukaryotic gene? This is a question. Now let us see the properties of introns. We can find answer for this question. Introns are mobile DNA sequences. DNA sequences is DNA containing adenine, guanine, thymine, cytosine. Mobile DNA sequences can move. They can splice themselves. Introns can cut and get detached, and the introns also attach to the specific target site. Introns can cut themselves and move out, and introns can join with another DNA. Introns act like mobile transposons like element. Transposons are movable genetic elements. Introns also act like mobile transposons. So they mediate transfer of gene between organisms. Introns can mediate transfer of gene between organisms because they are movable. Transfer of gene between organisms is called as horizontal gene transfer. Horizontal gene transfer occurs between prokaryotic cells. We have learned about horizontal gene transfer between bacterial cells in 11th standard. They are conjugation, transduction, transformation. Horizontal gene transfer also can occur from prokaryotic to eukaryotic cell and between eukaryotic cells. Horizontal gene transfer is now hypothesized to have played a major role in the evolution of life on Earth. It is believed that horizontal gene transfer might have played a role in the evolution of life on Earth. Horizontal gene transfer is possible because of the mobile genetic element nature of introns. Introns can move. Because of the movable introns, horizontal gene transfer is possible. This is the end of transcription. In the next part, we will learn about genetic code.